Okay. Anything.
Okay, it's 10:30 now. Good morning, everybody. Welcome to Saturday Matters. We have today a two-person act. Don McMillan is going to tell us about our brains, which we're all curious about, which we all use as much as possible and we know remarkably little about. So maybe we're going to find out more. Jerry is going to do the visual. A quick reminder, there's a bath. Want to read a poem, please, it's in the lobby of the Rainier Dining Room. Fill them out, and we'll find out who is the lucky person and persons who will have a poem drawn out of the basket and read it to us to celebrate National Poetry Day. I'm getting feedback here. Let me step.
There it is.
Away. Got it. Maybe Don will tell us a little bit more about his building security plans too. I had an idea. Maybe we could train some of the pets to be guard dogs. They could be. Some of them weigh as much as 5 pounds, I think. Help keep us safe. Okay, I'm going to shut up. We all want to hear what Don has to say. It's always worth our time. So go ahead and tell us about dopamine in the brain.
Thank you. As you see, I have some high-powered technical help this morning. And if things go bad, you can blame it on her. I don't have anything to do with the technology. Dopamine. Dopa. Sounds kind of dopey, I guess. At one time, dopamine was thought to be nothing more than a way station on the way to making norepinephrine and epinephrine. But more recently, it has been found to be one of the more interesting neurotransmitters in the brain. It runs a lot of important things, but when we get too much of it or too little of it, things can go bad in a hurry. So I'm going to talk a little bit about the good and the bad of dopamine. Now, some of this is going to be pretty technical, and I'm going to try to get everybody on the same plane to the extent that I can, but if it seems a little esoteric to you at times, remember that it's a whole lot more complicated than what I'm going to be talking about, because there are details that go on and on. So if I could have the first slide, please. I want to just talk a little bit about synaptic transmission. This is just a schematic drawing of a nerve cell. These are the dendrites, these kind of web-shaped things here, that receive incoming information from a previous neuron in the system. This is the cell body. This is the nucleus of the cell. This is the axon where messages go out when depolarization of the cell occurs. And these are the axonal endings where transmission goes on to the next cell across a synapse. And here's kind of a blow up of a synapse, and this is a pattern that's pretty common in most all of the nerves. The neurotransmitters are stored presynaptically in the axon. They're in vesicles to protect them. When the vesicle reaches the end of the neurotransmitter is ejected into this small cleft between the axon and the dendrite of the next neuron. It interacts then with a receptor. There are also various enzymes that are available to metabolize the released neurotransmitter, but in many cases, after it interacts with these specific receptors, it's taken back up into the neuron by an active transport process, restored again in vesicles and ready for reuse. So this is a a general pattern that occurs throughout the nervous system for transmission across the synapse. Could I have the next slide, please? We might have skipped one. Could you go back? Okay, that's it. To be more specific about dopamine, dopamine is synthesized in the neuron and then is released into the synaptic cleft, as we see here, quickly diffuses across this cleft, interacts with a variety of dopamine receptors, and we're not going to really be concerned about which receptors it uses today, so you can kind of forget about that. There is an enzyme within the nerve terminal that is responsible for the conversion of dopamine to inactive products, DOPAC. That enzyme is monoamine oxidase, and there's another enzyme outside the neuron, catechol-methyltransferase, that ends up with a final inactive metabolite, homovanillic acid. But a lot of the dopamine is, as we saw in that previous slide, taken up into the neuron, revesicalized, and ready again for reuse. Next slide, please. Well, where does dopamine come from? How does it get there? Well, it starts with an essential amino acid, phenylalanine, meaning the body can't make it itself. It's got to, I said enzyme, and that's not true, amino acid. The amino acid phenylalanine comes in through the diet and gives us this structure here. And then there are a series of reactions that get us to dopamine. Phenylalanine hydroxylase puts this hydroxyl group onto the ring. Tyrosine hydroxylase then adds a second hydroxyl group on the ring. And this is of some importance because this is what biochemists would call the rate-limiting step. That means it's the slowest step in the whole process, so the process can only go as fast as this enzyme will let it. We then put a second hydroxyl group on the ring, come up with a compound L-DOPA. L-DOPA is then decarboxylate, or given an L-aromatic decarboxylase enzyme to dopamine. And for today's purposes, we can stop right here because dopamine is the precursor of norepinephrine, but it stops in the cells that we're talking about. Why does it stop? It stops because dopamine beta hydroxylase is not present in these cells, so the end product then becomes dopamine. So that's pretty much how we get there. If I could have the next slide, please. I want to talk just a little bit about dopamine tracks. And what you're looking at is if the brain was cut right down along so. So you're looking at one half of the brain from the inside. And this is a schematic. The real thing doesn't look much like this. The nigrostrial pathway is not green, nor are these other pathways blue. And it also kind of looks as if this is a straight single neuron going back. But it's not. It's a pathway with thousands and thousands of neurons in it. It also looks like there's only one. But for example, this nigrostrial pathway, it's on both sides of the brain. So there are really two of them going back, one on each side of the brain. Now, we're going to concentrate on these two. There are generally four dopaminergic pathways that people talk about. The tuberoinfundibular tract, we're not going to pay much attention to that. That's involved with some hormones, particularly prolactin. The mesocortical tract, which goes up to the frontal cortex, we're not going to talk about that very much. That's pretty much involved with things like reasoning and that sort of thing. But we are going to pay attention to these two, the mesolimbic tract and the nigrostriatal tract. So next slide, please. So what does dopamine do in the central nervous system? Well, it mediates reward, and we'll spend a little time talking about that. It's responsible for the mechanism underlying the effects of some drugs. It's one of the mechanisms underlying the treatment of schizophrenia. It's very important in the initiation and modulation of body movements. Controls some hormone functions, but we'll pass on that today. And we'll also pass on its role in cognitive function. Okay, the reward circuit in brain. Reward circuit links a number of brain structures that control our ability to feel pleasure. Feeling pleasure motivates us to repeat behaviors. When the reward circuit is activated, individual cells in the circuit relay chemical signals. Some of these pleasures are largely innate: food, sex, drinking water, so forth. Other behaviors have to be learned as the organism grows. Money's a very obvious example. Money means nothing to a child under 5 years of age or so, but later it becomes quite an important reward that gives us a great deal of pleasure. And the neurotransmitter mediating reward in the brain is dopamine, which is released onto these dopamine receptors in the mesolimbic system of the brain. Now, if we didn't have pleasure, we wouldn't do things. The species would die. If eating food was not a reproducible event, the species could not survive. So we need to have a pleasure center. Next slide, please. Well, how did we come about learning about this? In the 1950s, a psychologist at the University of Michigan showed that if you planted an electrode in the median forebrain bundle of a rat, and that's just a subset of this mesolimbic dopamine system, the rat would press a lever to have a small electric current delivered to that median forebrain bundle. The stimulation was highly rewarding, and the animals repeated it obsessively. For example, a rat trained to press a lever to deliver mild electric current to the median forebrain bundle would just do this continuously to the extent that the rat would ignore food and water and would eventually stimulate itself to death. Later experiments showed that simultaneous electrical brain stimulation in this area of the median forebrain bundle and collection of the released neurotransmitter showed that the stimulation of the median forebrain bundle released dopamine. Later experiments in humans showed that electrical stimulation of this mesolimbic system in a variety of areas, not just to the median forebrain bundle, but others as well, produced intense pleasurable feelings. And it's now generally accepted that the reward system consists of a series of structures in this tract that we looked at previously, where the neurotransmitter is dopamine. It's the primary neurotransmitter mediating reward and pleasure in the brain. Next, please. Well, that's a good thing, but too much of a good thing is not always a good thing, because that's what happens with certain drugs. Methamphetamine produces its effects by releasing dopamine into reward centers in the brain. Could I have the next slide, please? So what is happening with methamphetamine is that it gets taken up into the dopaminergic nerve ending. It releases dopamine into the synaptic cleft where it interacts primarily with D2 receptors in this case. And some of that then gets handled by catechol methyltransferase, ending up as homovenilic acid, which doesn't really do anything, simply a product that then heads for excretion. The dopamine released in the synaptic cleft after it no longer binds to the receptor gets taken up by the active transport system. But if there's amphetamine still coming in, that gets released again. So the only way to turn this off is to have the metabolism of methamphetamine occur, such that the equilibrium between the dopamine in the brain and the blood vessels is disturbed, causing an outward flow. Go back, please, one slide. Cocaine blocks the reuptake in brain. Could we go forward again then, please, one? So here we're blocking this reuptake mechanism. So cocaine doesn't actually enter the nerve ending, but it acts to block the uptake of the released dopamine. So both methamphetamine and dopamine are producing their pleasurable effects by increasing dopamine binding postsynaptically, although they're doing so by slightly different mechanisms. And addicts will tell you that it's really hard to tell the difference between methamphetamine and cocaine as a pleasurable experience. Of course, they're generally able to do it based on the local anesthetic effects of the cocaine, which are not there with the methamphetamine. I think we need to go back here now on. Well, what about opioids? I've talked about opioids off and on over the years I've been here, and we know that their pleasurable effects are caused by binding to the mu receptor. And what's that have to do with dopamine? Well, it so happens that the mu receptor activation of those nerves then impinges on dopaminergic nerves. And so once again, we have dopamine being released, although not directly by an effect on the dopaminergic neurons. Fentanyl, heroin, etc. bind to the mu receptor, and then that then stimulates the release of dopamine on a different neuron, from a different neuron onto other receptors. There's lots of opioid receptors in this mesolimbic reward system. It's also known that other drugs that produce pleasure increase dopamine release, although the details of exactly how this is happening are not very well understood. The next slide, please. And the next one. Okay, let's switch over and talk a little bit about Parkinson's disease, because dopamine is heavily involved here. There are many symptoms, and we're not going to talk about all of them, but the kind of classical symptoms for severe Parkinsonism are a tremor, rest, bradykinesia, a lack of movement, and a great muscle rigidity. Well, what appears to be involved here is that other pathway. Can we find the? And now we need to go back further. Or forward, I don't know. My technical help is breaking down. So here's... Shall I quit now? So here's that Niagara's trial pathway. We're starting down here in this substantia nigra, progressing up here into the midbrain where the final synapses are, and this deteriorates. So now if we can get back to that other slide on Parkinson's instance. So the pathway from the substantia nigra to the corpus striatum in the midbrain, a major function of this pathway is to influence voluntary movement. The pathway both initiates and to some extent modulates activity in that motor pathway that controls voluntary movement. Now the pathway itself is cholinergic. There's acetylcholine is the neurotransmitter in the main motor pathways going down to the muscles and such. But this is kind of modulated, smooth, started and smoothed, if you will, by this pathway. And in Parkinsonism, particularly in the latter stages, there's a gradual decay of this nigrostriatal pathway, and that's what produces the symptoms of the disease, because you're losing this modulation. Next slide, please. Okay, how to treat it? Well, the classical way of doing this is to restore dopamine that's missing to the striatum. Well, one of the problems is the dopamine does not cross the blood-brain barrier. Now, it'd be nice if you could actually put electrodes in there and put the dopamine right where you want it, but that's not practical. So what you want to do is to give the drug peripherally and then hope that you're able to restore dopamine where it's needed. But the blood-brain barrier prevents this. Now, what's the blood-brain barrier? Well, at one time we thought this was primarily due to extraneuronal cells called glial cells that enveloped some of the blood vessels in the brain, and to some extent that may be true now, but now it's thought that perhaps more important are the very tight junctions in the blood vessels in the brain that do not allow certain chemicals to get through the walls of the blood vessels to get to the nerves. And that's very protective of the brain, because the brain is a very delicate piece of anatomy, and you want to protect it from poisons, from large molecules, from charged molecules, and that sort of thing. So we got to get some way to get around this problem. Well, L-DOPA, the precursor, remember, of dopamine, does go through the blood-brain barrier. Unfortunately, though, just giving the L-DOPA is problematic, because dopa decarboxylase is a fast-acting enzyme, and it will convert a lot of the dopa to dopamine before it can get through the blood-brain barrier. So the solution has been to combine the L-DOPA with a dark decarboxylase inhibitor, in this case carbidopa is the popular one, that converts dopamine. This will prevent L-DOPA from being, okay. What's going on here? I'm trying to think and read at the same time, which is not always possible for me. What happens is that you protect L-DOPA from the enzyme. The L-DOPA then crosses the blood-brain barrier, but the protection does not go through the blood-brain barrier. So you're keeping L-DOPA protected in the periphery, but when it gets to the brain, the protector does not follow it. So now dopa decarboxylase can form dopamine in the brain. Well, that works pretty well for a while. Some more a while being quite a long time, generally years we're talking about. But over time, it becomes less effective. And another problem is we're not pinpointing dopamine where it is needed. We're giving it to a whole bunch of areas in the brain, and there are a lot of dopaminergic neurons there as well, and that's responsible for some of the side effects, having excess dopamine in areas not associated with the nigrostriatal pathway. Well, something that's happened well after my time in talking about these things with medical students is that we have dopamine agonists. These are drugs that act like dopamine, but they're synthetic drugs that are given from outside. And I almost hesitate to try to pronounce these, but pramipexole, and how's the other one?
Ropinacol.
Okay, I'll buy that. Two of these have been developed as treatments for Parkinson's disease. So let's take a quick look at them with the next slide. We got the next one afterward. Okay, some advantages of these dopamine agonists. Well, they can cross the blood-brain barrier, so we don't have to do a lot of manipulation of them to get them up to the brain on their own. They don't need any enzymes or any special treatments to get to the brain. They act directly on dopamine receptors found on nerve cells, so they don't require any metabolism or modification, and they don't have to be stored in the nerve endings. They go right to that postsynaptic receptor and produce their effects there. They have longer half-lives, means longer duration, producing more dopamine receptor activation over a longer period of time than L-Dopa. It's become common to treat patients in the early stages of Parkinson's disease with dopamine agonists for several years before they get to the L-Dopa treatment. And this is good because it reduces exposure to levodopa, that's another name for L-Dopa, and therefore minimizes the impact of the side effects seen with L-Dopa, such as the dyskinesias that occur. When dopamine agonists are taken in conjunction with L-Dopa, they lower the dosage of L-Dopa required to smooth some of the effects of L-Dopa that are responsible for these late stages dyskinesias. Next slide, please. Well, a big question. Why does that nigrostriatal pathway decay? The short answer is we don't really know. Some patients have a history of head injury. Some patients share some genetic mutations, but the vast majority of patients don't have any of these problems. So we don't know. Now, something that I just discovered in the past week is some real interesting data on a compound called trichloroethylene, or TCE. It produces a 5-fold increase in the risk of getting Parkinsonism. Now, how do we come into contact with TCE?
That's cleaning fluid, isn't it?
Cleaning fluid. For years, it was used as the primary agent in dry cleaning. It's also used as a degreaser. It's used in numerous kinds of compounds and manufacturing processes. This is a big problem because, as you can see in the early part of that slide, in the past 30 years, the frequency of Parkinsonism has doubled, and projections are that it's going to double again in the next 20 years. Now I present TCE as a possibility. That's far from established at this point, but it is kind of interesting to speculate about it, that perhaps some sort of environmental toxin is responsible for the deterioration of the nigrostriatal pathway. But we really don't know yet. Next slide, please. Schizophrenia. Well, schizophrenia has been around for a long, long time. A very severe mental disease characterized by hallucinations, inability to deal with reality, sometimes with great agitation, etc., etc., etc. In the 1950s, Paul Charpentier, a chemist in France, synthesized a drug from a class of drugs called the phenothiazines, and the first of these was chlorpromazine. And early tests of the drug suggested that it might have an antihistaminic effect. The company that he worked for thought it might be useful as an adjunct to anesthesia. Early tests in mice, however, showed a remarkable calming of the mice when they were agitated and given chlorpromazine. In those days, we didn't have to go through all of the clinical trials. There were clinical trials, but the requirements were not anywhere nearly as rigid as they are today. So, two psychiatrists in France, Delay and Denneker, tried it in agitated schizophrenic patients, and they reported that it was pretty effective. Chlorpromazine and compazine became widely used in schizophrenia, and a number of other phenothiazines were synthesized, and many proved to be effective against schizophrenia. So subsequent studies on mechanism of action showed that the effectiveness of these drugs was due to the fact that they blocked dopamine receptors. The implication perhaps being that excessive dopamine might somehow have something to do with schizophrenia. Of course, it hasn't been that simple. Later, a class of drugs called the butyrophenones were synthesized, and the best known of these was haloperidol, the drug Haldol, a powerful blocker of dopamine receptors. And this really started a new era in the treatment of schizophrenia, because we didn't have anything that worked very well. Reserpine was somewhat effective in that it released dopamine and norepinephrine, had them quickly metabolized, but there were a lot of side effects with reserpine, and the phenothiazines and the butyrophenones were much more effective with fewer side effects than was the case of reserpine. Next one, please. Well, as treatment with these drugs occurred in schizophrenia, a constellation of side effects occurred. These included tremor at rest, cogwheel rigidity, bradykinesia, shuffling gait, etc. What's that sound like? Parkinson's disease, yes. This caused a very major problem. If you're blocking dopamine receptors, you're getting some of the same kinds of effects that one sees with Parkinsonism. And how do we control this? How are we going to keep people from being psychotic and yet not developing these so-called extrapyramidal symptoms? Titration was the major early example. For some years, when I was at medical school in Little Rock, I was also an advisor to the psychiatry department in the VA psychiatric hospital. And one of my first jobs up there was to review some of the physicians' prescribing habits. And you can see this very nicely if you looked at how they prescribed. Somebody would come in, they would go on a phenothiazine and continue on that phenothiazine while they were able to control the psychotic symptoms, but then the extrapyramidal symptoms would become quite debilitating, so they would ease off and go for a while, weeks, months perhaps, at a lower dose, no extrapyramidal symptoms, but a gradual reemergence of the psychotic symptoms. And so they just kind of be going up and down over a period of years. That was the better psychiatrists. There were a couple of them that when the psychotic symptoms got out of control, they simply added another one. Like maybe they'd be on chlorpromazine and they would add haloperidol to it. That's going to make things much worse in the long run. Now, one of the problems that came up during this chronic treatment with phenothiazines and butyrophenones was that if the dose was reduced or discontinued, an appearance of tardive dyskinesia. These are symptoms that occurred whereby patients would have a lot of facial grimacing. I used to like to do this for medical students, where they would do like so, chew on their tongue, bite on their tongue, have all these spasmodic movements. And unfortunately, when tardive dyskinesia occurred, it was hard to get rid of it. You could get rid of it by increasing the dose once again, but in the long run, that was probably going to make things worse because you were doing the thing that led to the tardive dyskinesia in the first place. That's still a problem in this area, these spasmodic involuntary movements. What do you do about it? Well, try to keep the dose as low as possible to keep it from occurring in the first place because it's hard to deal with. And then, of course, there are drugs that will suppress movement disorders to some extent, and that's about the only other way to go with the treatment of TD. Next slide. What about depression? Low levels of dopamine have been associated with depression. And indeed, the initial antidepressant drugs, the antidepressants such as imipramine, block the reuptake of dopamine. They also block the reuptake of norepinephrine and of serotonin. And this really hasn't panned out too well because it now appears that patients being treated for severe depression respond much better to drugs that block the uptake of serotonin than norepinephrine, the so-called SSRIs or selective serotonin reuptake inhibitors, rather than blocking dopamine and norepinephrine. Recently there has been some interesting things showing up, and I'm just going to mention this, I'm not going to go into it, but drugs that produce hallucinations, psilocybin and psilocin, for example, having some remarkable effects in some patients with severe depression. The mechanism of that, I don't know what that would be, but there are reports that in a certain segment of the patients that there are some very good effects with these drugs. Next slide, please. So I'm kind of coming toward the end here. Dopamine is now known to play many roles in the brain. And the dysfunction of brain circuits that use dopamine is involved in the development of many other disorders. But this is all at an early stage stuff, and I can't really give you mechanisms or even talk very intelligently about how reliable these data are. But Huntington's disease, anxiety disorders, obsessive compulsive disorders, attention deficit disorder, so forth, all appear to have some relationship to dopamine, but this is not very well described, and I really don't know how reliable those data are. Next slide, please. Well, I've only talked about dopamine in the brain, and I've only touched upon some of the actions of this drug in the brain, but dopamine biosynthesis occurs outside the brain, in peripheral nerves. Dopamine receptors have been described in the kidney, in the pancreas, in the lungs, lots of blood vessels outside the CNS. Renal dopamine is now being recognized as an important regulator of sodium electrolyte balance in the kidney, while defective renal dopamine production and or receptor function may contribute to the development of some forms of hypertension. And one more slide. Last slide, you'll be grateful to learn. That's the right one. Now, based on what I've said about dopamine, you might think that, dopamine's the most important of all of them, that everything runs on dopamine. Not true. Dopamine is important, but it's only one of those original big five of acetylcholine, norepinephrine, GABA, serotonin, dopamine itself. Since these early days on studying neurotransmitter function, numerous neurotransmitters have been discovered, and research is hard at work on their functions. It's now thought that there might be about 100 neurotransmitters in the brain, and these early ideas that we had that you could get all this complex brain function taken care of with just 5 neurotransmitters, probably pretty naive notion. There's still a great deal to learn about these more recently discovered neurotransmitters. Still, dopamine is a pretty important neurotransmitter in a lot of things that are necessary to our survival, and it's a pretty interesting and fascinating neurotransmitter compound, and I've only been able to touch on part of that story today. It's even much more complicated than what I've said. So thank you very much. Yes, sir.
Henry, okay, you and then you, okay? You turn the lights on? Yeah.
This
Yeah.
working?
Oh, sorry.
Is this working? Yeah, it is.
Yeah.
Yeah. I have two questions. One is, how does the blood-brain barrier discriminate to let some things through and not others? What kind of mechanism is that? And the other question is about, when did trichloroethylene become identified as a dangerous substance. It was a big improvement over carbon tetrachloride, which they used to use before that, I think. But I had quite a bit of exposure to trichloroethylene as a degreaser. And when was it determined that that was a dangerous thing?
Probably there were strong hints in the 1950s, and so it's been around for a long time, and its relationship to dopaminergic function is quite recent. Now, what was your first question again?
About the blood-brain barrier, how does it discriminate?
Well, it will not let charged molecules through.
Charged?
Any kind of a charged molecule through. And size is big molecules. Proteins, most proteins are not going to go through. They're way too big to get through.
Thank you.
So, well, one thing, the blood-brain barrier, they thought that more recently astrocyte foot processes had a lot to do with it too.
Astrocytes are a form of glial cells.
Yeah, and and at one time they were using estrogen to seem to ameliorate some cases of tardive dyskinesia, but that was a long time ago. So I don't know much about that. But the other interesting is Freud. I think I've read about Studies in Hysteria, the first book I had about Freud. But he published the first thing as cocaine as a topical anesthesia operating the eye. I think it was cataract surgery. That was the first thing published, one of the things published by him. And then he started using it and became addicted to it. But he realized it later in life that it was becoming an addiction. So late in life, he also had cancer of the hard palate, and he had to have a very painful, and had to have a wooden prosthesis so he could talk and swallow and so forth. And later in life, he decided that if he took the cocaine, he couldn't communicate with patients in therapy. So he stopped taking cocaine when he was talking to patients, even though he had this very painful cancer of the hard palate.
Yeah, cocaine and methamphetamine are very interesting. Certainly at the height of the high, they're very similar. But if you quiz addicts on who are very extensively exposed to all of them, they'll tell you that the cocaine high is probably the best. And if you set a monkey up with a cannula in the vein and with a syringe attached to that, and then have the syringe driven by a motor, and then give monkey a lever that will control the motor so that he can self-inject himself, how many monkeys out of 100 do you think will self-administer cocaine? 100, right? Not just the monkeys whose mothers were mean to them, or who grew up with a disadvantaged side of the railroad tracks. They all do it. It's an extremely powerful drug for self-administration and addiction.
Okay. Lowell, Jerry, and Al.
I was interested in looking back at Parkinson's. At one time it was thought that there was a cohort effect in Parkinson's related to the Spanish influenza. I'm wondering if there is any viral pathology suggested now, whether that was an old idea that disappeared.
I don't know the answer to that question, but we might have one back there.
He was claiming to have an answer?
Yeah, I really don't know of any viral connections that have been established.
And then Jerry, and then Al.
Yeah, early in Parkinson's disease, a lot of them were post-influenza. Actually, it was not the 1919, it was one about a year or two later in 1920 or 21, and they had a lot of post-encephalitic Parkinson's which come on just a few years after the attack. And but in recent years, that's not involved. But I had one patient that I saw when I was a resident that had another type of virus just something like 6 years before he came on, and had essentially post-encephalitic Parkinson. He didn't have any treatment, but a lot of rigidity. But none of them are, nowadays are thought to be due there. It's a polyfactorial thing.
Okay. You're next after Jerry, and then Fred.
So Don, I'm trying to just, because when I had kidney stones, the pain was relieved by giving me something wonderful that made me feel absolutely great. And I'm trying to understand, was that all due to just some little contact in my brain? I mean, the feeling was so widespread. It just felt glorious all over. Is it just that damn little thing?
Well, I hesitate to say that anything is all perfect and established. I can say that I've had the same experience with an opioid called meperidine that I had before an appendectomy. That you're ready to go into the OR and you're a little tense and upset, and this was in a small town, and one surgeon, and there was an automobile accident that kept me at the last minute from going in, and they put me in a room and gave me a shot of meperidine, and I thought I'm really a lucky guy to be here having this stuff, and I think at some level I knew that I better never mess with this stuff.
It's way too good.
Yeah, it's way too good. It just gets, and yes, I would say that certainly a large part of your experience is through the pleasure centers in the brain. The first part that I talked about.
Yeah, just going back to your first slide and showing the synapse, and began to realize how instantaneous this is. Because for a nerve pulse to go anywhere, it's going through millions of these perhaps.
Yes.
And I'm used to a chemistry lab where an experiment takes 5 minutes to resolve. This is instantaneous.
Yeah, if you look on the slides, it looks like they're pretty far apart. They're not touching. They're not touching, but they're really microns apart. They're very, very close, and that release and the whole system. I didn't go into what happens after it attaches to the receptor, but sodium channels open up and a whole bunch of ions flow, and they're second messengers like cyclic AMP and things like that that are involved, and all this takes place in an instant.
Whoever designed this was really on the ball.
What's all going on outside here? It's Fred's.
Don, in the field of speech pathology, there has been quite an interest in the difference between a physiological explanation of stuttering and a learned behavior of stuttering. But one of the things that was being explored was the effects of dopamine on the possibility of the starting of the motion that is necessary to produce normal speech, I guess.
Yes, that's interesting. We talked a little bit about Wishner's work at one time. His theory for stuttering was that the stutterer knows when the word is going to come that's going to be problematic, and knows before it happens and starts to build tension and anxiety as he approaches closer and closer to the word.
my recollection is they were referring to the research that was going on in this area as dealing with beta blockers. What's a beta blocker?
A beta blocker is norepinephrine has two major kinds of receptors onto which it attaches, one of which is beta receptors. They're very important in the heart. Beta receptors in the blood vessels cause dilation when they're interacted with catecholamines, and so beta is related primarily to norepinephrine and epinephrine. Did that answer your question?
I think so.
Similar receptors involved in alcoholism too, matter of fact. And I think I had a personal experience with the reward circuit. I think that led to my first slip.
Yeah, beta blockers were a very common treatment for blood pressure problems. They also were used quite a lot at one point for public speaking anxiety, that relaxation of the blood vessels would cause at least a peripheral reduction in anxiety.
But we don't need any of that stuff, do we?
I think,
No? Okay. Henry again?
about me.
Among the various abused, we might say, drugs, you didn't mention marijuana at all. Does marijuana have an effect on dopamine at all? Or?
That's not been studied very much. It's pleasures. It must have, but I don't really know that part of the literature.
Thank you.
Sorry.
We just have to keep testing it.
I think that Parkinsonism is a compensatable disease in Vietnam vets. And I'm wondering if that could be due to exposure to solvents.
Could be.
I think it's compensatable. I think it's on the list of things that Vietnam vets are able to get support for.
We just really don't know why that pathway gets in trouble over time. You would think the genetic aspect of it would be important, and perhaps it is in some subset. You know, we kind of look at diseases a lot of the time as if they're all the same, but they aren't. And that's probably true of Parkinsonism too, that there may be a lot of different reasons why this develops, but it's something that happens so slowly over such a prolonged period, it's really hard to get a feel for what's going on.
I can't hear you. You want to have a mic? Okay.
Just a quickie.
Just a quickie, is there any connection of any of this? Are they using it with epilepsy?
Epilepsy.
Not to my knowledge.
I wonder if you could just roll that back one slide.
Back one.
Yeah, just go back.
Something on
Just
this.
go back. You're okay.
Fall back.
Well, till he tells you to stop.
Just one slide